Okay, welcome to the speed run version of OTU, Ontario Tech University's Biochem 2 course. This is just be a series of lecture by lecture basis of a summarization of all the content in a short amount of time compared to a whole hour and a half lecture. So, let's start off with lecture 1, the most basic one, Principles of Bioenergetics. So bioenergetics pretty much just means the amount of energy utilized in a reaction, whether that's inputted or released, right? There's different variables associated with bioenergetics. This includes enthalpy, which is the release of heat or the need of heat for the reaction to occur. So if it's positive, it will require heat for the reaction to occur and is non-spontaneous. And if it's negative, then it'll release heat after the reaction will occur and will be spontaneous. Then there's entropy. Entropy is the amount of randomness in the reaction. If there's more enthalpy, then more it'll be a positive value and cause it to be spontaneous. If there's less enthalpy, which is a negative value, then it will be more ordered and non-spontaneous. And last one, the main important one, is Gibbs free energy value, which is delta G. Delta G is Gibbs free energy value. This value is used to determine the amount of energy released in a reaction or gained in a reaction or needed for a reaction. If delta G is negative, it'll be spontaneous. If it's positive, it will not. That's pretty much the basics, right? Those are the main key um, symbols used and variables used in bioenergetics. But let's dive into a little bit more. So to start off, you already know the relationship between Gibbs free energy and the amount of energy released, right? Bioenergetics, but you can also use bioenergetics for not only learning the spontaneousness of the reaction, but it can also be used to determine the direction of the reaction. So using another variable called the equilibrium constant, which is pretty much just the concentration of products over the concentration of reactants, which actually doesn't have any units, so you don't have to worry about it. Pretty much what you need to know, what's important, is if the equilibrium constant is greater than 1, right? Greater than 1, then that means the Gibbs free energy value will be negative, the reaction will be spontaneous and move in the forward direction. If the equilibrium constant value is less than 1, right? If it's less than 1, that means that it requires energy. So the Gibbs free energy value will be positive, causing it to be non-spontaneous, as known before, and then the direction will go in reverse, right? It'll go from products to reactants. If the key equilibrium is more than 1, it'll go from reactants to products, right? And then the last one is not very common, is if its K equilibrium is equal to 0, then the Gibbs free energy value is either plus or minus. That pretty much just means that is subject to changes depending on reactant concentration or product concentration. If whatever way they go, they can either go forward or in reverse. That's pretty much it. So now that we covered that, the importance of that, we can actually calculate the Gibbs free energy value using um, using enthalpy, entropy, and temperature. So pretty much your first equation that you get is Gibbs free energy value, which is delta G naught prime, which is at the standard condition. So um, at the same 25 degrees Celsius, which is 298.15 Kelvin, and then the same gas pressure, you can find Gibbs free energy, which is delta H minus T times delta S, right? Which is enthalpy. Entropy minus temperature times entropy gives free energy value. The next equation you can use is with using K equilibrium. when you have K equilibrium, you can find delta H. You can find delta H by um, negative RT, gas constant, temperature. So negative RT times ln of K equilibrium. Like I said before, K equilibrium is product concentration over reactant concentration. This way, it'll give you the um, Gibbs free energy value at standard condition. Now, an important thing to know about Gibbs free energy value is that there's two forms. There is the standard conditioning form, which is delta G naught prime, which is has constant temperature and constant gas pressure. And then there's a actual Gibbs free energy value. The actual Gibbs free energy value, which is delta G, is subject to temperature change, gas pressure change, concentration change, right? All these values will affect the actual Gibbs free energy value and the amount of energy either released or get needed for the reaction to occur. Now this is a different formula, and the key thing about this is also uses the Gibbs free energy value at standard condition. So the formula is delta G is equal to delta G naught prime plus RT ln K equilibrium. That's it. So pretty much ln RT ln K equilibrium plus delta G naught prime. After that is done, you have your actual Gibbs free energy. This is at either if it's in a human body at body temperature, which is 37 degrees Celsius, which is around 310.15 Kelvin. That's pretty much the generic actual Gibbs free energy temperature value. So pretty much that's it. That's bioenergetics, right? Bioenergetics uses all these variables to calculate the amount of energy released, calculate whether determine the direction of the reaction, determine the amount of heat released or needed, amount of energy released or needed, spontaneous, whatever the word is, spontaneity. I think I got it that time. So that's pretty much it for bioenergetics. But another topic that we covered in week 1 of lecture 1 of Biochem 2 is the actual reactions of like anything, right? Reactions of, let's say, glycolysis. So in terms of that, you need to realize that if you have a positive Gibbs free energy, right, whether it's standard or not, you need to realize that without any excess external source of energy, this reaction will not occur. That requires an additional reaction. This additional reaction is the reason why these reactions are additive. Because the reason why these reactions are additive is because you need a higher, or I guess a lower, more negative delta G value, a Gibbs free energy value, to complement or counterbalance the positive Gibbs free energy value that another reaction has. And then once you add one reaction, or maybe two, maybe 15, it varies, doesn't matter, they're all additive. Once you add all of these reactions up and you get a negative delta G value, then and only then will this reaction proceed forward, right? So let's give you an example. So we know ATP. ATP used in glycolysis, used in gluconeogenesis, used in variety of reactions in the human body. How do we make ATP? The simple, most basic version of this answer is you need ADP plus a phosphate group, ATP. That's it, right? Only thing is that this reaction is highly endergonic. What that means is it requires energy for the reaction to proceed in the forward direction. If you don't have energy, you won't have their product, which is ATP. This means we need to use an additional reaction which is highly negative in the Gibbs free energy value for this reaction to proceed to give off enough required energy to um, counterbalance the required energy and then allow it to actually move negative, right? So let's say we take phosphocreatine. Phosphocreatine is the phosphohagans we'll cover in lecture 3, I think, in the glycolysis lecture, where these are also phosphate molecules that release high levels of energy and have a highly negative Gibbs free energy value. Okay, so we have phosphocreatine. Phosphocreatine gets hydrolyzed, makes creatine in a phosphate group. This has, let's give it a number. This has negative 55 kilojoules mole Gibbs free energy value. And then you have ADP plus phosphate group to make ATP gives you a positive 30 kilojoules per mole Gibbs free energy value, right? So in this scenario, if you add these two, right, add these two reactions occurring, you will get a negative Gibbs free energy value of negative 15 kilojoules per mole, which thankfully allows the reaction to proceed in the forward direction in ATP, make ATP. The point I'm making here is using bioenergetics, using different reactions, we're able to create a product even though it has a the singular reaction by itself, the individual reaction requires energy. If you add it on with other reactions which have or exhibit a highly negative Gibbs free energy value, the reaction will still occur. That's pretty much it for bioenergetics. One last topic I want to touch upon is the topic of metabolism. So, like many of you know, like in Bio 1, Bio 2, I don't remember when, it's been a while, right? So, pretty much metabolism refers to the sum of all enzymatic pathways involved in synthesis and breakdown of molecules in the cell. Okay, so this is going to be real basic, but synthesis of molecules refers to anabolism, and breakdown of molecules, it refers to catabolism. So an important thing to know, which is very generic but comes back in biochem, right, which is important, is catabolism and anabolism. Catabolism uses carbs, proteins, and fats and makes smaller molecules, right? It degrades it from big molecules to small molecules via oxidation, and then or oxidation reaction, part of a redox reaction. And an anabolism refers to taking these smaller molecules like amino acids, sugars, fatty acids, and synthesizing larger molecules like proteins, polysaccharides, nucleic acids, right? Using a reduction. So these two reactions are vital for they're vital for glycolysis in terms of and vital for cell use, right? But an important aspect to know, right, is in these oxidation and reduction reactions, where are they getting energy from? This is a key component, and it's a similar, it's the same molecule in like everything. You've heard of it before. Is NADH, FADH2, and NADPH. NADPH we'll cover later on in a different lecture. It's a little more weird, but you have NADH and FADH2. These two molecules are pretty much the most basic electron slash energy carriers. They're electron carriers. They carry electrons from one reaction to another, so from an oxidation reaction to a reduction. An important thing to know is there's two forms of these, right? In a NADH, there's an NAD plus form and an NADH form. NADH form is the reduced form, and it holds the electron. NAD plus form is the oxidized form, and it accepts an electron. Same thing with FADH2. The only difference between that is a FADH2 requires two electrons to be fully reduced. So you get one FADH2, you grab it, FADH, great. It's half oxidized. You get a semiquinone. Then you go grab another electron, you get FADH2 fully reduced, right? So you have FAD plus and FADH2. These are two forms, right? And these are key players, key molecules used in all of the reactions: glycolysis, gluconeogenesis, glycogen formation, glycogen breakdown, right? These are used in lactic acid production, ethanol production, used in everything. And it's very important to know. That's pretty much it. That was a recap of lecture, actually 1 and 2. I didn't realize this until after I started looking at my notes. Lecture 1 and 2 focuses on bioenergetics and metabolism. It focuses on anabolism, catabolism for the metabolism portion. Bioenergetics focused on the variables associated with energy released and energy used in the reaction. I hope this helps you as much as it helped me. Thank you guys for watching. I'll see you guys next time.